Hello, everyone, and thank you for joining our webinar today, The Power of Chemical Characterization to Assess Changes in Your Medical Device. This is Alexa Tatarian, Chemical Characterization and Extractable Leachable Study Director here at Nelson Labs. And also joining me later in the presentation today will be Thor Rollins, our biocompatibility expert and consulting manager here at Nelson Labs. So today, you know, every test matters, and every test is important for our sponsors, and that's something that we really believe. And so today when we're going to be talking about changes made to medical devices, this is something that most medical device manufacturers experience. Changes happen to the device or the material, the processing, and we need to make sure that those changes are not adversely affecting biocompatibility or impacting patient safety in a negative way. And so every test matters, and we really want to help and give some information today that hopefully will be beneficial to go about accessing those changes and ways to set up a test plan. So the goals for manufacturers are to, manufacture biocompatible devices, and these guidelines and standards are outlined in the ISO 10993 standard. And any change made to a device needs to have biocompatibility reevaluated, no matter how major or minor that change is. And chemical characterization is a way to do that. And chemical characterization is using analytical chemistry to characterize the device and what is coming off of the device to try and predict the biological response of those compounds that we see. To talk about chemical characterization a little bit more, chemical characterization is the identification of a material and the identification and quantification of the chemicals present in materials or a finished medical devices. And I like to break this up into two groups. We can either do direct material characterization, where we are directly characterizing that device material, or we can do extractable and leachable testing, where we are doing an extraction on the device to see what leaches and extracts off of that device. This is an outline of the presentation and what we're going to be covering today. We'll talk about typical changes that manufacturers encounter or see that we've had experience here at Nelson Labs. We'll also review ways to evaluate the impact of these changes. We'll briefly cover some of the test options that are available to assess these changes. And then finally, we'll walk through some case studies, some real-life examples that we've seen and worked through with sponsors, and I think that'll be very beneficial for our viewers today. So there are two types of changes that we'd like to talk about today to medical devices. The one type of change can be process changes, anything changing with the manufacturing, production, or cleaning of that device. Have you changed the cleaning process? Have you added a new component in the process? Maybe you want to switch detergents, add a mold release agent, something like that. Or are you switching sterilization? Maybe you're using gamma and you're switching to ethylene oxide or steam, for example. Any type of process change in the production of the device needs to be evaluated. There are also material changes. So say you are working with a supplier and they all of a sudden go out of business and you need to find a new raw material supplier. Maybe you're adding an adhesive or another component to the device that needs to be evaluated. Maybe you want to make that device, manufacture it in a larger size. Any type of change to the material itself also needs to be evaluated for patient safety. To talk about process changes a little bit more, some questions to ask. Again, these are changes affecting the production, manufacturing, cleaning, sterilization of the device. Has there been anything new introduced in the process? Or has a step been added or removed? If a step has been removed, are the same potential residuals being able to be removed that were with the previous process? Or with sterilization, has there been an impact to the material at all? So anything new that's being introduced needs to be evaluated. Any change needs to make sure that nothing bad is being left behind or being newly introduced to that device. And it's important to always remember that every individual change will have a different impact regarding biocompatibility depending on what's being introduced, what's being taken away. Every change should really be evaluated separately. With material changes, so these are changes to the device itself or a raw material for that device. Has the raw material changed? Has there been a new component added to the device? Maybe you just have a new supplier, but that polymer or metal is the same, or they say it's the same. You know, all of those changes, again, need to be evaluated. And with material changes, you know, with process changes, we're often easily being able to assess those changes with analytical chemistry. We can maybe target that new potential compound to see if it's being left behind with some residual analysis. Maybe if a step's been removed, we're able to, you know, really narrow down what has changed to try and see the impact. With a material change, that does have the potential to maybe have more of an impact on patient safety, as that really does have that direct contact with the patient. And so sometimes with a material change, you may actually need to do some additional biocompatibility to make sure that that change is not causing harm to the patient.
I wanted to step in real fast and add a little bit more about the impact of a material change and how to assess the material change of biocompatibility. Like Alexa just mentioned, the material changing of a device can have the biggest impact as far as biocompatibility is concerned. And how we assess that really depends on the type and the amount of the material changed in the device. With a biocompatibility test, we calculate the surface area of the patient contacting portions of the device and then extract those portions based on the surface area. So the larger surface area a device has, the more volume of fluid we add to extract. So therefore, if your change to the device is in a very small portion of the patient contacting surface area, then that impact to the overall toxicity of the device is going to be very small. That means that the assessment can be done very easily. We can look at it through some screening, some chemistry, but very minimal amount of testing might be needed because of that change being so small on the surface area. If we are changing the majority of the material that contacts the patient, well then we have to do more of an assessment because now we're impacting more of the patient. If your change is applied to a non-patient contacting portion, an assessment should still be done, but ISO 10993 is very clear that the biocompatibility is only for those portions of the device that is patient contact. The assessment of a non-patient contact may include, though, during processing, for example, sterilization, is there any transfer from the non-patient contacting portions to the patient contacting portions of the device? So that may include some chemistry to evaluate leachables that may be different on the patient contacting portions of the device pre and post the change to those portions that are not in contact with the device. So as you can see with the material change, there are some thought processes that you have to step through as far as the impact of the surface area of the change, if it's to the patient or non-patient contacting portions, or would one of the processing impact the patient contacting portions to a non-patient contacting portion change. These are some of the questions that we walk through when evaluating the material changes and some of the endpoints that we address when making an evaluation.
So just to summarize some of the change impact discussion that we've been talking about today, you know, changes can be unexpected, and manufacturers have to evaluate them as they happen, as they occur. Changes can also be strategic decisions. Maybe you're a manufacturer who's trying to improve the process. Maybe you can cut down resources and time on the cleaning procedure by making some changes. Same with the device. Maybe there's a newer adhesive out there that's going to benefit the patient by having it on there, or improve the function of the device by adding that in there. Again, any change needs to be evaluated. And so with your original device, you know, that went through the FDA submission or whatever regulatory agency you were working with. That original submission and that device that was tested, if anything is changed in the processing or material of that device, it needs to be evaluated. And again, anything like an addition or a removal of anything in the process needs to be targeted with testing for that impact. This is a brief summary of test options that are available for direct material characterization and extractable leachable testing. We won't go too in-depth of these test methods, but we wanted to briefly cover them to make you aware. FTIR, or Fourier Transform Infrared Spectroscopy, is a test for identification of a material. It can be very helpful. DSC, or differential scanning calorimetry, characterizes thermal properties of polymers. GCMS, or gas chromatography mass spectrometry, is a quantitative and qualitative analysis of more volatile and semi-volatile compounds. These can be either compounds that are leaching from a device material itself or residuals left over from manufacturing that could leach or extract out of the device over time. LCMS, liquid chromatography mass spectrometry, again, qualitative and quantitative test for non-volatile compounds. ICP-MS, inductively coupled plasma mass spectrometry, quantitative metals analysis test. There's also physicochemical testing, which analyzes physical and chemical properties of polymers. And then, of course, there's cytotoxicity, which is one of the most sensitive biocompatibility tests. Anytime there's a change made to the device, we definitely recommend at least repeating cytotoxicity, which should have been completed in that original submission, for a quick check to make sure that things are looking all right. In our experience, we've seen over 90% of the time, if there's a failure with some of the biocompatibility tests, that we do see that failure show up with cytotoxicity. And so a great sensitive screening test to include in the test plan to look at that impact to biocompatibility and patient safety of the change. And quickly, just some additional information about the test methods and the process. Again, there's direct material characterization methods that are, directly characterizing that device material. And then there's also extractable leachable testing that's more of an extraction. An extraction on the device is performed to look at the compounds that are either left behind on the surface of the device or leach from the device material itself. With extractions, we either typically extract simulated use, more clinical use conditions of that device, or we can extract more exaggerated or worst case conditions. Maybe this would be more appropriate for devices with more prolonged permanent exposure. We may want to elevate temperature for extraction, use harsher solvents, things like that. With also testing, we're usually doing comparison testing here when we're evaluating the change. I mentioned that the original device that went through that submission, that data is already on file and should have had the biocompatibility completed. So when we test this new device with a change, if you have that baseline data or samples from the original process to compare back to, that can be really most helpful in evaluating the change to see if those changes are, in fact, beneficial to implement, if there are less or fewer compounds or fewer quantitative values of those compounds left over time. I do want to mention, best case, when we do the comparison, the devices look similar in testing. We're seeing similar compounds extracting out. We're able to see a similar configuration of the device material. But it is a possibility to see differences, especially if you are changing the device material itself. And so in that case, if we do see differences, often a toxicological review needs to be performed on those compounds that are found during testing to still assess if those compounds are safe and at acceptable levels for the patient. So for example, if we did do testing and we did see a significant amount of a new compound that didn't show up with the new device, additional risk assessment may need to be performed to take a look further at that quantitative value. And at that point, there still could potentially be some additional biocompatibility tests that need to be performed if we do see differences. So something to keep in mind and mention there. Now I'm going to turn the time over to Thor to walk through some case studies of examples that we've had experience with recently working with change evaluations.
Thanks, Alexa. So we have 5 case studies here. I will be talking through the first couple of them, and then I'll turn it back over to Alexa to handle the last one. But On these are real-life examples of devices that have been through a change that we use a combination of biocompatibility and chemistry to help and justify the change to be able to assess the safety of the device, the new device. In this first case study, we had a catheter system. The catheter system went from having a clear plastic catheter with the ink on the outside that was manually applied. They are now looking to change the catheter to a frosted shaft with the ink embedded into it during the mold release process to help eliminate a step. So the ink is on the outside still of the catheter and does not have intended patient contact, but we still want to assess to see if the ink could migrate through the plastic into the fluid path of this device. The fluid path administers liquid, saline, to the patient, and so you have saline running through the catheter into the patient. And we want to assess the change of the frosting that's incorporated internally into the plastic and also the ink. Once again, both of these changes were not intended to have patient contact, but we were worried about migration through the plastic. So what we did is we performed a side-by-side chemistry comparison. As you can see on the right side of the screen, we performed chemistry analysis on the original device and on the new device, the one with the changes. Then we looked at the types and amounts of compounds coming off of each type of device. We compared those types and amounts to one another. And what we found is actually the frosting process limited some of the leachable profile of the catheter device. We actually had less compounds coming off than we did in the original. And the ones that were coming off the frosted, the new device, were the same types and same amounts as the original device. So you look at it as a whole concept through all these chemistry evaluations, we actually proved that the changes improved the leachability of the new device, and that did not impact the overall biocompatibility of this catheter system. Case study number 2 also had to deal with a material change, which happens to be one of the most common evaluations that we run here at Nelson Labs. With this material change, we were changing a material in one device that was being used in another device that the manufacturer actually made themselves. So we had a distal end of the device that the manufacturer had already made with a previous device or a device on the market, and they were just changing this other device with that already approved material. Now, that may seem like no big deal, but there might be there has to be evaluation done to see if there's any processing impact to changing those materials, even though the materials were the same. For biocompatibility, we just don't care about materials, we care about the processing, which Alexa talked about earlier. So even though you're using the same materials, you have to evaluate to see if the processing is the same. In this case, when we looked at the processing, we had similar processing in both the devices that we were comparing. So we have a similar material that the change is going to, similar processing that the change is going to. So the assessment included cytotoxicity to analyze that risk. As Alexa said, cytotoxicity is a great screening test. It's not a cure-all, but it's a great screening test to evaluate impact of a change. And in this case, since we were able to justify that the materials themselves were the same, the processing was the same, the cytotoxicity was a great reference. We want to put this case study in here to show chemistry is a very powerful tool. Sometimes when you're evaluating it, chemistry may not be necessary to evaluate a change. It depends on the type of change and the materials and processing that are impacted to be able to evaluate that change. Case study number 3 was actually very interesting. In this one, we had 2 changes. We had a distal tip to a device that was going through a change, and then the handle was also going through a change. Now, normally a handle is not intended to have patient contact. In this one, the handle actually does contact the skin of the device, of the patient. So the distal end had more of a tissue contact, more of a more severe contact type, where the handle just contacted the skin itself. So there's less of an issue as far as safety to the patient, but still need to be addressed. So for the handle, we actually looked at the material that the change was going through, and we analyzed the material and realized that the material themselves and the compounds released from that material were not sensitizers. There was no sensitizing impact to that change. So in this case, we were able to run a cytotoxicity and irritation to be able to evaluate the concern with the handle, with the skin contact. And the sensitization was justified by using the chemistry and the material evaluation of the device. With the new component in the distal end of the device that goes into the patient, the new component actually had full biocompatibility data, including implantation for its own material. So once again, we need to worry about processing. In this case, it was off-the-shelf material that they were just adding to the distal end of the device. Very minimal processing, but we still wanted to assess that, so we used cytotoxicity to run an assessment on the combination of the previous material with this new addition. Now, we treated this as two separate studies. We treated the distal end of the device that has the unique type of contact, which is the tissue, and then the handle, which has the skin contact. We treated them separately in this case study. The fourth case study has to do with a Class 1 device. And as many of you are probably aware, and those maybe are not, a Class 1 device are 510K exempt for the FDA, which means that they do not technically have to submit a 510K for approval to put a device on the market. I always tell everyone about Class 1 devices, you might be 510K exempt, but you're not safety exempt. So there are two potential impacts to not being safety exempt. Just because you're Class 1 doesn't mean that you're not still going to have a negative impact to the patient. There's a recent case of a wristband that was a medical device, it was a Class 1, that they did not perform any biocompatibility testing on because it was a Class 1 device. Well, the patients in contact with this wristband started having a sensitization reaction to the material. Now, this created a recall, a new analysis of the material, a very expensive and long impact to the company. So just because you're not needed to submit to the FDA does not mean that you still have to address safety. Chemistry is a great way to address the impact of a change also for a Class 1 device. In this one, we had a stainless steel tip of a device that contacts the skin for a very short period of time. They went from a manual hand polishing step for the stainless steel to an electropolishing process. The same compounds were involved, just one was a manual hand process, the other one was in a machine. But just to make sure that there wasn't an impact in that change, because the electropolishing process is a change, even though you're using the same material, we were able to run a chemistry evaluation and a cytotoxicity to help evaluate the impact of that change to the device. Once again, a Class 1 device, so the risk is pretty low, but we're still able to show that the same residuals that happen on the manual hand cleaning process was happening on the electropolishing process to show that there was no impact, and the cytotoxicity helped tie everything together. Now for case study 5, I will turn it over to Alexa.
Thanks, Thor. So for this last study, we were working with a client that had an implantable device that had contact with cerebrospinal fluid contact. And they were changing sterilization. They were currently using gamma, and they were switching to ethylene oxide. And so this client wanted to be conservative in their approach because it did have cerebrospinal fluid contact. It wasn't implant. They wanted to be conservative in analyzing this change. However, they did not feel that they needed to repeat all of the biocompatibility that was completed with the original submission. So what we ended up doing here is a full-blown, very robust chemical characterization profile comparing the devices from each sterilization method. We compared both implant devices. And so we did some baseline characterization with FTIR and DSC on the materials, as well as a volatile, semi-volatile, and non-volatile compound analysis. We also looked for metals, and phthalates or plasticizers were also a concern for one polymer that was present on this device. We also did a quick cytotoxicity test. We included particulates as well as a full surface cleanliness analysis as well. And so this client wanted to be a little bit more conservative in their assessment, and they also were interested in having this data on file and completing it with this change so they could have it as baseline data for any future changes that are made to the device. And that's actually something that's really beneficial with chemical characterization data if you're willing to make that investment up front or with a change that has been made. You now have that data on file And anytime a future change is made to the device, you have that baseline data to compare back to with that chemistry data. And so that can be very helpful moving forward if you do decide to make that investment to have that data on file for future changes. So there's a lot of future benefits available with chemical characterization as well. So in conclusion, chemical characterization is a great tool to use to address changes made to the device, either the device material or processing as we have discussed today. And it's also important to note that every change is so unique and has a different potential impact on the patient depending on how major or minor, where that change is occurring. And so to end here today, if you are making a change or come along the line down the road where you do have to make a change to your device, we would love to help you evaluate that change, walk through it with you, try to come up with a test plan. We have a consulting group at the lab that specializes in just that. So thank you so much for joining us for our webinar today. If you have any questions, please let us know.